As bioimaging has always been a strong aspect of research at BOKU, we would like to show you how amazing imaging projects can be. The range of samples that can be visualized with our microscope is quite broad. Bacteria, yeast cells, cell cultures, tissue slices, isolated organelles, plants or parts thereof, artificial materials, worms, early-stage embryos, and many, many more. We will show you 3 different projects that benefited from the new microscope and hope to be able to share our excitement about the new possibilities for conducting research with it.
Our recent research focuses on how plants respond to and adapt to drought stress. When a plant senses drought stress, it coordinates its activities via the plant hormone abscisic acid, or ABA. And here our hypothesis is that a group of endosomal trafficking receptors, the toll proteins, function in the degradation of the ABA receptors, thus terminating the signaling cascade. So in one of our experiments, we used transgenic plants stably expressing, on the one hand, our protein of interest, the toll proteins with a green fluorescent tag, and on the other hand, the ABA receptor with a red fluorescent tag. In this experiment, we want to assess if the two colocalize in the stomata of plant cells. We used confocal laser scanning microscopy, where we focus the laser on a very defined area and get the emission of the fluorescent only from this defined area. For this purpose, we performed Z-stacks where we optically dissect our specimen, so there's no physical dissection. It's really just with a laser we go through the different layers in the Z-axis, and then afterwards we can compile these images to a 3D image where you can very nicely see the resolution of the two proteins. So on the one hand in green are toll proteins, in red the ABA receptors, at a very nice and high resolution. If we remove the toll proteins, for example, by knocking them out, we have prolonged signaling in the stomata, resulting in extended closure of the stomata and therefore increased drought tolerance in these plants. We want to assess if the same effects are true for crop plants, such as barley and soya, and also investigate if there's no negative trade-off between this enhanced drought tolerance and undesirable traits such as reduced yields. This research will contribute to the breeding of more climate-resilient crops.
One of the interests in our group is the production of recombinant proteins of pharmaceutical interest in diverse plant tissues. The purpose was to improve the host, in this case Nicotiana benthamiana, it's a plant similar to tobacco, so that it would produce a higher amount of the protein of interest. The protein synthesis takes place in the endoplasmic reticulum. That's a large organelle that stretches over the whole cytoplasm. The strategy was to enlarge the size of the cell where the proteins are produced and folded. Let's say like this, if we have a larger factory, we expect to be able to produce more product. What we can see here very nice is this network of tubules that constitute the endoplasmic reticulum. You can see very nicely the dynamic of this organelle. It's continuously being remodeled. We can see these tubules forming this network. Sometimes we have larger areas that would correspond to a so-called cisternae, and we can have also one element that defines the endoplasmic reticulum are these empty spaces that are called polygons. The size of the polygons, the balance between tubules and cisternae, the diameter of these tubules, these are all characteristics that define this network and help with the determination whether we have achieved this enlargement of the endoplasmatic reticulum. What we have done here, we take pictures of the non-modified plant and the diverse mutant lines that we have generated, and we take several pictures. Via image processing, we can establish the density of this network in each of the samples, so control against mutant, and then we can determine if we have reached or not this increased density. I have to say that we indeed managed to increase and enlarge this density, and this was very easy to see under the confocal because the balance between tubules and sheets, so these larger areas, moved towards the cisternae, which means that they were more abundant, and the size of the polygons in the mutants was also smaller, indicating that this network was much denser.
My research focuses on the plant secretory pathway, a production platform for proteins and polysaccharides. I am particularly interested in the basic cell biology of the Golgi apparatus, which acts like a post office for the cell. It receives, processes, sorts, and sends materials off to the correct location inside or outside the cell. Each Golgi stack resembles a stack of pancakes, and each pancake represents a flat membrane-bounded compartment called cisterna. I am showing you now leaf epidermal cells of a plant called Nicotiana bentamiana. My plants simultaneously co-express two different fluorescent fusion proteins, both of which are located in the Golgi apparatus. One is a menosidase, whose gene sequence has been fused to that of a green fluorescent protein, and the other is a sialyl transferase that has been fused to a red fluorescent protein. Both enzymes are located in different parts of the Golgi stack. This spatial separation can usually be seen in two-dimensional images acquired by the confocal microscope, as you can see it here. But every now and then we have a situation where the fluorescent signals overlap. We can acquire highly resolved images of a Golgi stack along the Z-axis, and this stack of images can be visualized using a 3D software tool. Here, for example, you can see that the two fluorescent signals are clearly shifted here in this Golgi stack. Whereas here, when we look at the Golgi stack from the top, the two fluorescent signals appear to co-localize. But when you look at the three-dimensional Golgi stack along the Z-axis, you can see that the red and the green signals are shifted. Over the years, we have identified ways to move N-glycan processing enzymes between cisternae, which has allowed us to modulate the N-glycosylation pattern and produce glycoproteins with tailor-made N-glycans in Nicotiana bentamiana. This glycoengineering platform is now being used by research and industrial institutions worldwide to produce therapeutic proteins. This is a good example of how basic research can be successfully translated into application.
Please do not hesitate to contact us if you think microscopy would be of any help to your project. This technique can be a game changer. You will literally see your research object from a different angle. Visit our webpage, check our infrastructure. I'm looking forward to seeing you in the Core Facility Multiscale Imaging.